Okay, so our next topic is going to be on transcription and translation, or another way to talk about it is we often refer to the whole process as gene expression. Right? How do we go from DNA, I have it in the red box over there, DNA to mRNA, or messenger RNA, we'll talk about that a little bit more, to proteins. And so this whole process of gene expression involves two separate processes, or the conversion of DNA to RNA, which is called transcription. So we transcribe genes into messenger RNA, and then messenger RNA becomes translated into polypeptide or protein sequences. And so what we're going to talk about for the next number of slides here is how the molecular processes that are involved in taking genes and transcribing them into messenger RNA, and then taking that messenger RNA and translating it into a protein. And of course, it is this gene expression that affects the function of cells. It determines, we've been talking about genetics now for the longest time. So how do those genes, or those different versions of genes, we've been talking about those alleles, how do those actually affect the phenotypes or the traits that we see? Well, those genes get expressed through transcription and translation and affect what's going on in the cell. And so let's look at a specific example of that in the next slide. So the first process we want to talk about is transcription. So transcription is basically RNA synthesis. So what you're doing when you transcribe a gene is you're taking the DNA sequence and converting it into an RNA sequence. And it's that the transcribed region that contains all of the information that we need in the messenger RNA to then make a protein. And in this slide, you see a number of other important parts of a gene that are involved in transcription. And so transcription begins at a place along the DNA called the promoter, and we're going to talk some more about that. There's the transcribed region, or the region that has all the sequence that's important for making the protein. And then there's a terminator, which tells the RNA polymerase, or the enzyme that's synthesizing the messenger RNA, it tells it where to stop. And then you also see in green here at the beginning, there's often a number of other kind of regulatory sequences. So how do cells, know when to transcribe this gene? And so we're going to talk some more about that when we get into gene regulation. But this is just an overview of what we're talking about with transcription. So it's using the DNA sequence from a gene to synthesize an RNA sequence. And again, there are a number of important other sequences that are involved in this. And let's just look quickly at an overview of how this works in prokaryotes first. So this slide, remember with prokaryotes, they don't have a nucleus. And so you can do transcription and translation kind of all at the same time. So you have DNA that gets transcribed into RNA, and then that RNA can get immediately translated into proteins. So there's no transport in and out of the nucleus as one would see in eukaryotic cells. So let's look more specifically then at transcription in a prokaryotic cell. So this slide shows the three primary steps or stages that are involved in transcription. And this is specifically showing you some of the proteins that are involved in transcription in prokaryotic cells. So we can divide it into three stages, again, as indicated here. So the first stage is initiation. So how does the cell know where the genes are and where to begin this transcription process? Well, first it searches for these promoter regions that I showed on a couple slides back. And in prokaryotes, there are some very important proteins that are binding to these promoter sequences. And one of them is called sigma factor. And you can see that as this blue blob here, binding to that blue promoter region on the DNA. So sigma factor can bind to the promoter region, and then what it does is it signals the RNA polymerase to come in and bind to the DNA and begin synthesizing the RNA or transcribing the gene. And so that is kind of how initiation works in prokaryotes. In eukaryotes, it's very similar. There are proteins that bind to the promoter region and then enable or bring in the RNA polymerase to bind and kind of tell the cell where to begin transcription. But in eukaryotes, it's not just sigma factor. There's a whole slew of transcription factors. There are many, many, many of them. And we'll talk about a few when we get into gene regulation a little bit more. But basically, there are a number of proteins that bind to the promoter and then signal the RNA polymerase to come in and begin transcribing the RNA. That's basically the summary of how initiation works. And then the second step, shown here in number 2, is what we might call elongation, or the actual synthesis of the RNA transcript, or of the RNA. And so the RNA polymerase moves down the DNA, and it basically copies one of the DNA strands into RNA. So remember, with DNA, A always paired with T, G always pairs with C. But remember, there aren't T's in RNA. So as you're transcribing messenger RNA, the template DNA strand, that's what we call the strand that's being copied into messenger RNA, synthesizes the RNA based on A pairing with U. So wherever an A is found in the DNA, a U is put in for the RNA, and then we also have G's and C's pairing between DNA and RNA. And the RNA synthesis always, again, just as was true with DNA synthesis, it occurs in the 5' to 3' direction. So we have the elongation or the synthesis step. And the RNA polymerase continues down the template strand of DNA until it encounters a specific sequence called a terminator. And once the terminator is reached, this causes the RNA polymerase and other association factors to sort of disassociate, and the completed RNA transcript then also dissociates from the DNA, and transcription is completed. So this is just a basic overview of how it works. This particular diagram shows prokaryotic transcription, but the 3 steps, initiation, elongation, and termination, are the same in eukaryotic cells. Again, it's just that there are a number of additional proteins that are involved with the initiation step, but basically it's very similar between prokaryotic and eukaryotic cells. But remember, eukaryotic cells have a nucleus, and so there's an additional step after transcription is completed that occurs in eukaryotic cells. And let's just look at that quickly in the next slide. So this slide shows eukaryotic gene expression. And again, remember, all the DNA is inside the nucleus. And that whole process that I showed in the previous slide of transcribing the genes into messenger RNA, all of that occurs inside the nucleus. So we have transcription from DNA to RNA. And so then the messenger RNA actually gets transported, has to get transported outside the nucleus, because translation actually occurs out in the cytoplasm. And this is not the case in prokaryotes, right? Because there is no nucleus in a prokaryotic cell. So you don't have that additional step of something being transported outside the nucleus. So there's one other step that is specific to eukaryotic cells that I want to talk about next. So we have transcription, or the conversion of DNA into RNA, but you notice here on this slide there's an additional step that's called RNA processing that occurs in the nucleus before the messenger RNA, mature messenger RNA, leaves the nucleus to begin translation. So let's talk about what this RNA processing is. And again, this only occurs in eukaryotic cells. It does not occur in prokaryotic cells. So there are 3 separate processes that are involved with the conversion of what we might call a pre-mRNA to a mature mRNA in eukaryotes. And all three of them are shown here. We're going to go through each one in a little bit more detail in the next few slides. So we have our gene that's shown here in red across the top. There you have your promoter and you have your terminator, and then you have what we might call the structural gene between the beginning and the end there. And we have a couple of things shown in red there called exon 1, 2, and 3, and there are green introns 1 and 2. And we'll come back and talk about those in a little bit. So we have transcription, right? So the promoter signals the beginning of transcription, and a messenger RNA or an mRNA is copied from the template strand of the DNA. And before that, pre-mRNA becomes what we call a mature mRNA and is actually transferred outside the nucleus into the cytoplasm. Three things happen. So one of them is capping, one of them is splicing, and one of them is tailing. And you see those three here indicated by those three arrows. And all three of those occur to give us a mature mRNA from the pre-mRNA. All right, so let's talk about each of those separately in the next slide. One of the first things that happens to the messenger RNA before it becomes mature and leaves the nucleus in eukaryotes is what's called 5' capping. So you see in the top here you have a yellow single-stranded RNA molecule, and this kind of unique and interesting nucleotide, a G nucleotide, literally gets glued onto the 5' end of the messenger RNA. So it gets this little cap or this interesting unique base added to the 5' end. And we'll talk about what the role of all of these are once we get through all of this. But 5' capping is one of the RNA processing steps. And then 3' poly-A tailing or 3' tailing is another processing step that occurs. So we get a unique nucleotide added to the 5' end of our messenger RNA. And then the other thing that happens is a whole string of A nucleotides are added to the 3' end of the messenger RNA to convert it from a pre to a mature messenger RNA. And both of these things, both the 5' capping and the 3' tailing, really help and assist with the export of the messenger RNA from the nucleus out to the cytoplasm. They help stabilize the messenger RNA through that transport process so that the messenger RNA doesn't become degraded. And then the other thing we'll see once we get to translation, especially that 5' cap, it really helps the translation machinery bind to and recognize a mature messenger RNA so that it can begin translation. So 5' capping, an interesting nucleotide gets added to the 5' end before it leaves the nucleus, and then a whole string of A nucleotides are added to the 3' end, again, before it leaves the nucleus. And then the third process is what's called splicing, and so let's look at that in a little more detail in the next slide. So in splicing, what happens is there are a number of what we call non-coding regions or introns that are part of the pre-messenger RNA or pre-mRNA. And those in this slide are shown in the green. There are a number of segments that are in the DNA that are then transcribed into the pre-mRNA that get removed. In the conversion of the pre-mRNA to the mature mRNA, those green sequences or introns are spliced out and removed, and then the exons, or those expressed sequences labeled exon 1, 2, and 3 there, are joined together or are linked together to form then the mature messenger RNA. So a mature messenger RNA is capped on the 5' end. It has got a poly-A tail on the 3' end, and all of the introns are removed. And all of this occurs before that messenger RNA leaves the nucleus and moves out to the cytoplasm for it to be translated. And so what are some of the reasons why the splicing occurs? Let's talk about what we know and understand about that in the next slide. So in this slide, we're looking at the phenomenon called alternative splicing. Basically, what alternative splicing means is that you can have more than one way to splice a pre-mRNA, depending on the cell type, depending on maybe the time during development. So if we look at this diagram here, so again, the introns are shown in green. Those are the pieces that are eventually spliced out, and then the introns are mostly yellow, but you can see intron 3 and 4, one's purple and one's pink. So if we follow those, if we look at the splicing pattern in nerve cells, the arrow to the left, what you notice is number 4 acts as an intron because it's missing. You only have 1, 2, 3, and then you skip to 5. But then if you look at the splicing pattern of how the exons are spliced together in muscle cells, you see 3 is missing and 4 is put in place. So what alternative splicing basically means is you have a single gene that can transcribe a single pre-mRNA, but then that one pre-mRNA can get sort of cut and pasted and spliced in different ways depending on the cell type. Again, one way in nerve cells in this diagram and another way in muscle cells. So this is kind of letting genes sort of give more bang for their buck. So a single gene giving rise to 2 different mature mRNAs without having to have 2 different genes. You have a single gene, but you get 2 different messenger RNAs. Again, they're not really different because most of the exons are the same, but there are some slight differences in the coding region between those 2 mature messenger RNAs that both arose from the same pre-mRNA. So what's the advantage of that? So what we noticed when we sequenced the human genome, before we understood and had sequenced all the genes in the human genome, there were a number of people who predicted, based on what we know about how many proteins there probably are in the human body, how many genes do we think we have? And many of the estimates were upwards of, 50,000, 100,000 different genes. Well, we now know that those estimates were very high because in reality, there are only something on the order of 25 to 30,000 genes. So we way overestimated how many genes there were. And one of the reasons why we believe there are fewer genes in the human genome than we would expect is because this process of alternative splicing is very, very common in the human genome. And so let's look at some numbers in the next slide. So this slide is showing a number of different organisms, from E. coli or bacteria to yeast, and then Caenorhabditis elegans is a worm. We've got flies, and then Arabidopsis thaliana is a plant, and then humans are at the bottom. And again, a little bit of a description of the complexity is there, and then it gives you the size of the human the different genomes in millions of base pairs. And so you can see, of course, the human genome is the largest of all those that are listed here. And the number of genes that we see, actually this plant, Arabidopsis thaliana, has more genes than humans do. And so how do we explain that? How can we have so few genes but have so many different proteins to give rise to a fairly complex organism like humans? Well, if you look at the column that's indicated there with the red arrow, the percentage of genes that undergo alternative splicing. So remember on the previous slide, so this is the percentage of genes that when they're transcribed into a single pre-mRNA, those pre-mRNAs can be spliced in many, many different ways to give you different protein sequences or different proteins from those alternatively spliced messenger RNAs. And look at this column. So clearly humans have a very high percentage of alternatively spliced genes, upwards of 70% of the genes undergo, when they get transcribed, they undergo this alternative splicing. So 70% of human genes actually can give rise to multiple proteins from that one single gene. And so this is one of the reasons why we believe that our original estimates of how many genes there were in the human genome was much, much higher than what we came to know and understand, that there are on the order of 25,000 human genes. And so why splicing? Well, one explanation for it might be because you have the opportunity for alternative splicing, which makes the gene sequences, or the DNA, to be, especially in the case of the human genome, to be more efficiently and effectively utilized, because you can use alternative splicing as a mechanism to get more proteins, as opposed to just having many more genes to give you many more proteins. Okay, so we've talked about transcription. There's one other thing that I want to highlight that I mentioned before, but it's really clear to highlight in this diagram. So we're talking about transcription, converting DNA into RNA, into messenger RNA. And I have circled here two terms, the coding strand and the template strand. I mentioned the template strand before, but let me highlight this again before we move on and start talking about translation. So the DNA is double-stranded, right? But we only need one of those strands to make RNA, because RNA is single-stranded. And the DNA strand that's used to copy into RNA is called the template strand. So we always call that the template strand. And right, DNA, the two strands of DNA are complementary to one another. So the complementary strand to the template strand is what we call the coding strand. And why do we call it the coding strand? Because if you look here in this diagram, and you look at the coding strand of the DNA, which is the top red one, and you read that from 5' to 3', ACT, GCC, etc., and then you go down to the yellow strand there, which is the actual messenger RNA sequence, and what you notice from 5' to 3' is that the sequence of the messenger RNA is the same as the sequence of the coding strand of the DNA. It's just that you have U's in place of T's. And so that's why the strand of DNA that is complementary to the template strand is called the coding strand, because that is actually the sequence of the messenger RNA, just substituting U's for T's in the RNA. So I just wanted to highlight that. Okay, so we use the template strand of the DNA to copy into RNA, a pre-mRNA in eukaryotic cells that undergoes a number of processing steps to give us the mature messenger RNA. In eukaryotes, then that gets transferred out of this nucleus into the cytoplasm, where we can start translation, which is then converting this RNA sequence into an amino acid sequence. And there's a lot of important things on this slide, and we're going to go through and describe and define all of them here. But this is a good slide to continue to go back to, because there's a lot of information here. But let's now start talking about translation, which is, again, taking that RNA sequence, the mature messenger RNA sequence, and translating it into the order of the amino acids that we're going to put together to get a particular protein. Before we get started into kind of the molecular details of how translation works, it's important that we talk about this table here, which is the genetic code table. And certainly I don't want you to go through and memorize all of this, but it's just, it's a nearly universal table that gives you the information on the messenger RNA, which is indicated here in 3 nucleotide letter codons, right? So in the top left, UUU, UUC, UUA, etc. Those are sequences from the messenger RNA. And what you can get from this table is, if you have the sequence UUU in a messenger RNA, the amino acid that codes for is the amino acid indicated here with PHE, which is phenylalanine. So let's look at another one. If I go down to the bottom left, if I have a codon in the messenger RNA that has the sequence GUU, what that means then is the amino acid that's going to be coded for is VAL or valine. So this is almost like what you want to call like the decoder ring, if you will. So this is the sequence of what we call 3 nucleotide long codons that are found on the messenger RNA that establish or that then are translated into particular amino acids. And if you count up the number of codons on this table, there are 64 codons. So there's 64 different ways that you can put sets of 3 nucleotides together, 4 nucleotides in sets of 3. So, and we also know that there are only 20 amino acids, right? So you have 64 codons, but they code for only 20 different things. And so that's why, again, if I draw your attention to the bottom left, you see that there are actually 4 different codons that all code for the amino acid VAL or valine. GUU, GUC, GUA, and GUG. All of those codons will translate into the amino acid valine. And you also see highlighted here in white, you see the codon AUG, where it says met or methionine is that amino acid /start. So that's the start codon. That is always the first codon that is translated. We'll talk about that more in a minute. And then there are 3 other codons shown at the top, UAA, UAG, and UGA. And next to them is written the word stop. And so those are the 3 stop codons, and we'll also talk about those in more detail shortly. So this is the universal genetic code, or the universal relationship between the RNA sequence and the protein or the peptide sequence. And so this is an important decoder table that we need if we want to look at a messenger RNA sequence and figure out what is the primary sequence of the order of the amino acids in a particular protein. Okay, so translation then is that conversion of the RNA now into protein. And so we see a simple diagram here again with some important sequences indicated for us. So in the previous slide, I mentioned the start codon, which is shown here in green. So that's the codon, AUG codon, that specifies for the very first amino acid. So there's always a start codon near the 5' end of the messenger RNA, where the translation or the sequence of the protein actually starts. And then if we look on near the 3' end, we see that red box there indicating a stop codon. And that is always one of those 3 codons that were on the previous slide, indicating that's the end of translation. So that's where the sequence of the protein should stop. And then, of course, between that start codon and the stop codon are a whole series of codons that specify various amino acids. And this is what gives us then that primary sequence. If you remember back to when we talked about protein structure, the primary sequence of a protein is the order of the amino acids. So from the start codon all the way through the coding sequence to the stop codon. And then you see just in front of the start codon, there's a little area, or this is a sequence that's not translated, but that's the 5' end of the messenger RNA where the ribosome, which is important for translation, will physically bind to that messenger RNA. And that's another important sequence to keep in mind when we start talking about how translation works. So what I want to do before we go through the steps of how translation works is I want to talk a little bit about the main players, or sometimes referred to it as the translation machinery, because there's a lot of players in this. And so let's talk about those first and define what their role is. And then we'll talk about how translation works as a process. But let's talk about the translation machinery first. So one really important player in translation is what's called the transfer RNA or the tRNA. Okay, so this is, it's not a messenger RNA. it is an RNA molecule, and you can see in the top diagram here, it's a single-stranded piece of RNA, but because of the sequence of the RNA, it actually folds on itself. So it complementary base pairs with itself, forming this interesting three-dimensional structure. And so what is the transfer RNA? The transfer RNA is, if I was to use an analogy, and we think about translation, and we think about translation being sort of a place to manufacture something. So if you think about the ribosome, which is sort of the manufacturing center that we're going to talk about here in a minute, and then the messenger RNA is sort of the directions or the instructions, the blueprints of what you're manufacturing. I would say the transfer RNA is sort of like the delivery trucks that are bringing in all of the supplies that we need to build our protein. So the transfer RNA is kind of that intermediary between the messenger RNA and the protein. So the transfer RNA carries the amino acids into the manufacturing center to start building this protein. And so there is a 3-nucleotide sequence. You see it at the bottom of both of these diagrams here that's called the anticodon. So we have codons on the messenger RNA, these 3 base sequences, and then we have anticodons on the transfer RNA. And the codons on the messenger RNA and the anticodons on the transfer RNA are complementary to one another during translation. And so we'll see that when we start talking about the mechanism. So that's at the bottom of these transfer RNAs. And then towards the top, we see an acceptor stem is what it says there, a 3' single-stranded region that is the acceptor stem. And that's an acceptor for an amino acid. So amino acids are literally attached to the transfer RNA, and the transfer RNA then sort of, we might think about as it kind of gets loaded up with various building blocks or various amino acids. And so let's take a second to talk about how that works, because there are enzymes that specifically link the transfer RNAs that have a very specific anticodon to a particular amino acid so that they can bring in the right building blocks into the ribosome. And this enzyme that's important for attaching the amino acids it's a mouthful. It's called the amino acyl tRNA synthetases. So we have a little bit more detail on how those work in the next slide. So let's look at that. So this slide gives you kind of a step-by-step process into how the tRNAs are what we call charged. So tRNAs, remember, they get loaded up with a specific amino acid to eventually bring that amino acid into the manufacturing center. So this big tan or brown blob in this slide is your amino acyl tRNA synthetase. So these are enzymes. And so what do these enzymes do? They bring the amino acids, which are shown here as a blue ball, in contact or in proximity with the tRNA, and then with the help of some ATP hydrolysis, they link the amino acid to a specific tRNA. Okay, so you have an amino acid that comes in to the enzyme, the synthetase enzyme, and you have a specific tRNA that it gets brought in, and its specific anticodon is recognized by this enzyme, and then the amino acid literally gets linked or charged onto the end of the tRNA, and then those tRNAs are released out into the cytoplasm to then partake in translation and participate in translation. So you have a specific amino acid joined physically to a specific tRNA. So in other words, you're kind of loading those delivery trucks up with very specific building blocks or specific reagents that you need to build your protein. So we've got our tRNAs that transfer the amino acids to the specific spots for building the proteins. So the tRNA is one important part of the translation machinery. And another really important part is where all of this takes place, which is the ribosome, which we've mentioned before. But let's look at the ribosome now in more detail. So So here we have the structure of a ribosome. It's shown in sort of a more molecular detailed picture at the top and a little more broadly at the bottom. And so the ribosome is basically a complex of two different subunits. There's a large subunit, which is the one on the top, and then in the top diagram, it's sort of that purple and gray one, and in the bottom, and on the small subunit is the one on the bottom that's in those two different colored blues. So you have a large subunit and a small subunit of the ribosome. And you can see the messenger RNA, shown in the bottom diagram here in yellow, physically attaches to the small subunit. And then the large subunit is where those charged tRNAs come and sit into a number of different sites in the large subunit of the ribosome. And again, we're going to go through this in some detail, but I just want to introduce you to sort of the overall structure here. So you see there are three different sites labeled E, P, and A in the large subunit of the ribosome here. The E stands for the exit site, P stands for the peptidyl site, and A is the aminoacyl site. And each one of those sites in the large subunit plays an important role in translation. And so we'll see the role they play here in a minute. But the primary players in translation, or the translation machinery that we need to keep in mind, is the transfer RNAs, or the tRNAs, that specifically get charged or attached to amino acids, and then the ribosome, which is composed of two subunits. And the ribosome is really where all of the components of the machinery come together. The messenger RNA comes together with the instructions, the tRNA brings the amino acids in, and the ribosome sort of coordinates all of the efforts. Okay, so now we have some idea of all the players here. So now let's look at a general overview of the translation process. So just like we saw with transcription, we can divide translation into the same three steps. There's an initiation step, an elongation step, and then a termination step. And this slide shows just an overview, and then we'll look at each one in more detail in some subsequent slides here. But remember, in eukaryotes now, we are out in the cytoplasm. The messenger RNA that was first made through transcription in the nucleus has been processed, capped, poly-A-tailed, spliced, and moved out into the cytoplasm. And so all these ribosomes are out in the cytoplasm. And again, if we're talking about prokaryotic cells, the messenger RNA was transcribed or made, and then translation happens right away because you don't have to, there is no nucleus, so you don't transfer anything out to the cytoplasm. So if we just quickly look at an overview here of translation. So at initiation, what do we have coming together? We have the messenger RNA joining to the small subunit, and then a transfer RNA attached or charged with a specific amino acid here, shown with that blue ball, and all of this comes together with the ribosome. So all of these things come together, and the start codon is placed in that P site at the beginning, or in the ribosome. And then through elongation, the messenger RNA gets kind of slid through the ribosome, and new tRNAs are brought into each of the sites, and new amino acids are added and joined together to form a protein. And then in the end, termination occurs when a stop codon on the messenger RNA moves into one of the sites on the ribosome, signaling that this is the end of the coding sequence, and then the ribosome, the tRNAs, and the protein that's made all sort of dissociate. The messenger RNA then can go associate with another ribosome and begin the whole process, but we have now successfully translated and synthesized a protein. So we have initiation, elongation, and termination, but now let's look at these in a little bit more detail. So we'll start with initiation. So during initiation, again, the small ribosomal subunit binds to the messenger RNA. So you have your yellow messenger RNA binding to the ribosome. And how does it physically bind to the ribosome? Well, in eukaryotes, it's that 5' cap that helps the ribosome know and recognize that this is a messenger RNA. And in prokaryotes, it's a little bit different. The recognition is a little bit different. The messenger RNA sequence binds to a different RNA that's part of the ribosome. But nonetheless, there are specific recognition components that are part of the messenger RNA, whether you're talking about a prokaryote or a eukaryote, that allow the small subunit of the ribosome to physically bind to the messenger RNA. And so the messenger RNA then binds to the small subunit of the ribosome. And the start codon is moved into the P site of the ribosome. So you can see the start codon there in green in this diagram. And so that start codon then binds to that initiator tRNA. So remember, if we go back to the genetic code table, the start codon is always AUG. So the start codon has that sequence, and so the complementary anticodon is found on that initiator tRNA, and then that brings in the amino acid methionine. And once that initiates, the large ribosomal subunit then binds, and that tRNA is put into the P site. And so we have an empty E site and an empty A site, but the synthesis of the protein has now been initiated by a tRNA with its amino acid bound to the start codon. So as translation moves into its the second stage here, the elongation stage, what we have, as I said just previously, is we have an empty A site. So that leaves a codon, a 3-base codon on the messenger RNA is sitting there at the base of the A site. So the tRNA with the complementary anticodon comes into the A site, and with it, it brings its particular amino acid that has been attached to it. And so then we have tRNAs. In step 2 there, we have tRNAs attached to specific amino acids in both the P site and the A site of the ribosome. And those two amino acids then are in close proximity to one another, and a peptide bond is formed between those two amino acids. And with that, the tRNA that just attached its amino acid leaves through the E site. The whole thing shifts down, moving the tRNA that was in the P site, or that was in the A site, to the P site, and again now opening the A site for the next tRNA to come in. I always find that this particular step of translation is hard to visualize in this sort of static way. So on Canvas I have put at least one video that is, I think, a good one that for you to watch on how translation works. I think this is a lot easier to see and understand when you watch it sort of in motion. So basically we've got a new tRNA coming into the A site. The amino acid between the amino acid that's in the P site and the A sites get joined together. So the tRNA that's in the P site then leaves through the E site, and the whole thing shifts down, moving the one from the A to the P, and now the A site is open and available again for the next tRNA to come in. And which tRNA comes in, it depends on what the codon is, because then that matches or complementary base pairs with the anticodon on the next tRNA. And this continues as the ribosome moves down the messenger RNA in that 5' to 3' direction, and continues to join amino acids as those tRNAs keep bringing those building blocks into the ribosome as dictated by the codon sequence that's in the messenger RNA.